being
do that. Hi, everybody. So I had a question, and I want to go through that. I did post an answer on the class website. The question asked me to go through the different types of synapses, such as exodendritic, exosomatic, probably dendrodendritic, exo axonal, things like that. So the first thing we need to do is understand that simple pattern of something, oh, something. And you're already well familiar with this. Hold on, let me make sure this is not going to be turned. I might need to flip my camera. Yeah, I think I need to flip that. Hold on. Okay. Oh, you can't see the questions. Really? That's too bad. So here's what I see. Hold on, let me share a screen. That'll probably help. Oh, by the way, we have breakout rooms, I think. Yeah, I can do the bare minimum. Okay. Documents not published. Okay, thanks. That helps. I can fix that. Let's see. That's real simple to fix. Cool. I'll have to fix this for all of them, but anyone on the internet can view this. Cool. All right, so now refresh it. Let's see how it looks. Hopefully you see something that looks like this on my end. Any luck? Maybe I didn't do the right document. Still seeing no's. You know, that was the quiz. Yeah, okay. Hold on 1 second. Give me 5 seconds. Anyone on the internet should be able to view this now. Okay. Okay, so if I share the right one, I think that fixes it. Thank you, Morgan. How about now? Have you refreshed your browser?
I can see it on mine. Excellent.
All right, so it'll slowly get there. All right. Anytime I make something new, we always have some kinks to iron out. Okay, so blank, oh, blank. So the question asks about the different types of synapses. So we need to get this pattern. The pattern is from O to. So you've heard of the corticospinal tract. This is a tract that's going to run from the cortex to the spinal cord. How about the Cortico-Thalamic tract? That one runs from the cortex to the thalamus. We also have, as you might imagine, a thalamo-cortical tract. And that tells you that it runs from the thalamus to the cortex. So we're telling you location as well as direction here with this. And the same thing is true for these types of synapses. The most common synapse is going to be axodendritic. Axon to dendrite. This is telling you about the flow of information, generally speaking. Of course, there are retrograde messengers that can allow back communication, but the typical flow of information is going to be from the neurons axon to the dendrites of its postsynaptic partner. And this just has to do with the surface area. So most of the neurons' receptive surface area is going to be on the dendrites, just because they have a whole lot more surface area than the cell body. If I were to try to draw this a little more anatomically correct, usually the cell body is going to be about this size. I have a big dendrite coming off, lots of branching. There'll be several dendrites coming off. Every neuron's a little different, but your typical pyramidal cell here is going to have one large apical dendrite, several basal dendrites. All these are going to have very fine branches coming off of them. They'll have spines. Those are going to increase the receptive surface area. But consider the dendrite surface area versus that of the cell body. There's just a whole lot more of it. So this is your bread and butter synapse, from axon to dendrite. Now, where we form that synapse is going to allow us to kind of guess at what kind of synapse it is. Is it excitatory or inhibitory? Now, the good news is I have to tell you. So you'll know if it's an excitatory synapse because I'll tell you it's an excitatory synapse, or it's a glutamatergic synapse. If I tell you it's a GABAergic synapse, it's inhibitory. Typically, let me simplify the neuron here. We're going to find our inhibitory synapse. The inhibitory synapses are going to be in this general area. So they'll form on the more proximal dendrites as well as the cell body. Now, the reason for this is that this location allows the inhibitory synapses to have a stronger effect, so that we can inhibit the neuron when we release our inhibitory neurotransmitter. Because where we tend to find our excitatory synapses, let me do another color. Excitatory synapses are going to be found more distally. And there's a good reason for this too. The dendrites are not uniform in diameter. They're going to taper as you move away from the axon. So while I've drawn it as a straight line, that's just a shorthand. Really, the dendrites are going to have a decrease in diameter as you move further away from the cell body. So if we were to plot it, here's your cell body, and this is far away, the diameter of your dendrite is going to decrease. So that tapering of dendrites decreases their diameter and increases their internal resistance. Ohm's law is really important. Please know it. Now's the time. Go ahead and know it now. V equals IR. So out here in the distal dendrites, compared to the proximal dendrites, we have much higher resistance. So the currents that we generate, because we have higher resistance, will create greater potential changes. So we're more likely to excite voltage-gated ion channels that are out there in the dendrites. So it makes good sense to put your excitatory synapses further away where you have higher resistance, and to put your inhibitory synapses on path. So when that charge tries to make it to the cell body, we can block it. So these locations make good sense. There's going to be some intermingling. You'll still get excitatory synapses on proximal dendrites, but the general rule is that the further away you go, the more likely it is to be excitatory. Because an inhibitory synapse out here at the tip of the dendrites isn't going to do anything. It's just a waste of space. You need to have them on proximal dendrites or even on the cell body. So when you hear axosomatic, this is another one that you see in cartoons. This is going to be inhibitory. Axodendritic, it depends. Where is it? They can be excitatory, they can be inhibitory. You'll just have to be told. Now, if they're forming on dendritic spines, so let's zoom in here, that section of dendrite can have little spiny protrusions called dendritic spines. The name makes good sense. And they have variable morphologies. Some will be a nice mushroom shape, some will be stubby. There's a short stubby one right there. Some will be long and thin. Notice we're tapering. The synapses that form here are going to tend to be excitatory. Most of them are. So in adults, which we are all adults here, what you almost always see is a single excitatory synapse on dendritic spines. On rare occasion, they're inhibitory. At least we think that because that's what they look like in an electron micrograph. And sometimes you actually see a spine split into two. So you can actually get two synapses onto a single spine. But for the most part, dendritic spines, axospinous synapses, those are going to be excitatory. The most common, axodendritic, it's a mix. If it's on spines, think excitatory. If it's on proximal dendrites, think inhibitory. If it's on distal dendrites, think excitatory. But I'll tell you. So these are the most common ones, and you're going to see these in any basic neurobiology class. There are, of course, synapses that form on axons. Those would, of course, be called so from axon, axonal synapses. So in this case, we're forming a synapse on the axon itself. So let's get a new neuron up here. The axodendritic and axosomatic synapses are going to change the membrane potential, and they're going to determine whether or not this neuron fires an action potential. So when we fire our action potential, that's going to travel down, and we're going to spit out neurotransmitters. So all the activity out here in the dendrites is going to sum in the cell body and collect at the initial segment of the axon. And if we excite the neuron, it fires an action potential. And if it fires an action potential, it then communicates with its postsynaptic target, whatever that is. Now we can affect whether or not we're going to fire an action potential by forming axonal synapses. We could do that at the initial segment of the axon, so the axon initial segment right here. This is where we generate an action potential. So if we inhibit the neuron here, that trumps everything else going on here. It doesn't matter if you depolarize the cell enough up to the point of the cell body. If you inhibit it at the axon, you can then prevent the firing of an action potential. So these are going to be inhibitory. We could also form synapses with the presynaptic site itself. So we can form synapses here and affect how excitable the presynaptic terminal is. So we could increase its excitability, prolong its depolarization, and increase the amount of neurotransmitter release. So if we increase neurotransmitter release, we say we facilitate it. So the message that's sent out from this neuron now becomes a little bit stronger as a result. It stays depolarized a bit longer, more calcium, more neurotransmitter release. More on this, I think, in lecture 5. Not 100% on that one. But in this first unit, we're going to talk about neurotransmitter release. We can also inhibit it. That's the other option. We can also decrease the amount of neurotransmitter that gets released by narrowing that window of depolarization, reducing the amount of calcium that comes in, and thus reducing the amount of neurotransmitter we spit out. So the message will be not quite as strong. these are not quite as common as your exodendritic and axosomatic synapses, but they exist. You should know that they exist, but you probably won't think about them much after this. But this is a way that other neurons can affect how well this neuron is communicating with its target, because that's what it's all about. Neurons are here to communicate. And the way that they communicate is by generating electrical signals within themselves, propagating that down the axon, and then spitting out a chemical messenger. That's usually going to create some sort of electrical event in its postsynaptic target. It could be another neuron, it could be a muscle, it could cause contraction. So we can affect them in the dendrites, the cell body, or even the axon. Now we also have, for the sake of completeness, dendritic synapses, and that's exactly what it sounds like. Synapses between the dendrites. These could be chemical. More commonly, what you're going to see are electrical synapses. So a couple of neurons near each other. This is in the back a little bit, but they line up their dendrites and they form electrical synapses. And that's going to synchronize their activity. So you'll see this between two or more neurons, so that when one is active, the other tends to be active. They share their cytoplasm, so when one depolarizes, so it has an positive movement of the membrane potential, so does its neighbor at the same time. So they're both going to communicate with their targets, and usually they're going to go target some similar location. So rather than acting just as an individual, now we have a chorus of neurons who are sending the same signal at the same time. They're either all stimulating or all inhibiting their target, and they'll do it together because they've linked together their dendrites. So when one is excited, they all get excited. I think that basically covers it. Were there any other questions?
What did you say at the end there, where the two of them, like where there's an increase in one, there's an increase in the neighbor?
Yeah.
That's dendritic?
Yeah, if they're forming gap junctions between one another. You can also have somatosomatic synapses where you put your somas together.
Okay.
So in those cases, if you're forming electrical synapses, what you're doing is synchronizing your activity. So let's just imagine some electrical synapses here. So we'll have two cells, and they're gonna kind of butt up next to each other like that, and they'll form electrical synapses. And those are actually just holes in the membrane. Now, of course, there's proteins lining them. And they form what we call gap junctions. So they're connections between cells that have a little gap in them, a little space, so that we get free exchange of cytoplasm, both directions. So these cells can effectively act as one. So they can share a variety of things. They can share RNA. They can share glucose. They can share, in this case, charge. So if this neuron here, and it'll have some dendrites coming off of it. These connections could also be between dendrites. It's just easier for me to draw the cell body here. When this cell starts to have a buildup of positive charge, that'll float on over and increase the positive charge in this cell, so that whenever one reaches threshold potential and fires an action potential, so does its neighbor. So they're going to act as a unit. We'll put our neighbor right over top. So when one is excited, and spits out neurotransmitter from its axon, so is the neighbor. And so what you're essentially doing is doubling up the chemical message that you send wherever it is you're communicating to. So this is a way of essentially creating an amplifier, so that instead of having one neuron try to whisper some message, you have a group of them shouting it.
Thank you.
You're welcome. Alrighty. So volume transmission and peptide transmitters. Okay. I suppose the harm that we might be thinking of is maybe a prolonged increase in intracellular calcium that then, of course, leads to death. So we're not in that window. All right, so the question here is, how do we release these, and how do we do so safely? So how are we releasing peptide transmitters, which need a burst of activity, and how are we releasing neurotransmitters away from a postsynaptic site without then causing harm? Have I mentioned extrasynaptic receptors before? I think last semester, maybe they were mentioned. When glutamate escapes the synapse, it can stimulate extrasynaptic receptors, and those are generally toxic. They cause loss of the synapse or loss of the neuron. So we're not going to use volume transmission with glutamate. We're only going to see volume transmission with those neuromodulatory transmitters, like dopamine, for example. So we can release dopamine away from the postsynaptic site and allow it to diffuse over a large volume to hit a great number of synapses all at the same time. That's safe to do because we don't have toxic dopamine receptors like we do for glutamate. So that's what will make that safe. So whenever we're talking about volume transmission, all we mean is that we're not talking about a synapse where we have just a little 20 nanometer gap between our pre and postsynaptic site. Instead, the site of release might be further up in the axon, maybe here. And so when we spit out the neurotransmitter, it has to diffuse some distance to encounter a synapse. Maybe it's here, I don't know, maybe it's over here. But it's not immediately opposed to the site of release. So this would be volume transmission. But we still have our good old presynaptic sites here. We're releasing classical transmitters. They're really small vesicles, and they're filled up usually with little amino acid derivatives, something like that. Or they could be amino acids themselves, like glutamate or glycine, or they can be derivatives, like GABA, dopamine, norepinephrine, things like that. So we start with an amino acid, and then we modify it a little bit. Most transmitters are going to be that. We also have another type of neurotransmitter that are peptides. These are stored in larger, what we call dense core vesicles. Dense, in this case, refers to how the core of the vesicle looks. So it's exactly what it sounds like. When we say dense, what we mean is it looks dark in an electron micrograph. And that's because it's filled up with protein. And that's all that's telling us. Just like our postsynaptic density is called out because it's a dense region at the postsynaptic site. The reason that it looks electron dense is because it's filled up with protein. Your postsynaptic density is filled up with proteins. It's filled with them, and those proteins would, of course, be neurotransmitter receptors, scaffolding proteins that hold them in place. In this case, we're filled up with little bits of proteins called peptides. So little, I don't know, 3 amino acids, could be 13 amino acids. Not a full-blown protein, but a string of amino acids, not a single one, like we'll see over here in our standard synaptic vesicles. The dense core vesicles are going to be filled up with peptides, and they're going to be kept away from the active zone. So right here, this is called the active zone. We'll talk about this later. This is where we have calcium channels. So we're going to get a burst of calcium here, and it'll diffuse away a little bit. Each time we fire an action potential, we bring in more calcium. It diffuses a little further away. So whenever we fire a burst of action potentials, we keep building up calcium until eventually we have enough calcium at extrasynaptic sites to initiate release. So a single action potential is going to be enough to get release at the active zone for classical neurotransmitters like glutamate, GABA, your monoamines. But in order to get release at extrasynaptic areas for our peptide transmitters, we need a burst. So if we think about the calcium levels within the cell, each action potential is going to cause a little burst of calcium. And as you pile them up on each other, you're going to increase intracellular calcium levels. Not to toxic levels, though. We're not there. And this is going to be very quick. So we're not keeping the neuron active for many seconds or things like that. Bursts will terminate. So we have safety mechanisms in place. How do we do it without causing harm? We have potassium channels that are stimulated by calcium. So when we build up enough calcium, these potassium channels activate and hyperpolarize the neuron, so it terminates the burst. So potassium saves the day. That's how we don't cause harm. But we need this buildup of calcium so that it can diffuse far enough away and stimulate neurotransmitter release. As we will find out in, I think, lecture 5, it might be 4, I don't know. I think it's 5. Neurotransmitter release is all about bringing calcium into the cell. So wherever you bring calcium into, you could release neurotransmitters if you have vesicles there. So we have them docked at the active zone right next to our calcium channels. So as soon as the action potential arrives, we spit out neurotransmitter. If you want to release neurotransmitter further away from there, you need a burst. Now for volume transmission, we of course have calcium channels here. So when the action potential propagates down, we get a little burst of calcium and we get neurotransmitter release. That just then has to diffuse away. For volume transmission, I want you to be thinking monoamine, dopamine, norepinephrine. Don't think about glutamate. That would be a terrible thing because we have toxic extrasynaptic receptors. Not the case here. Is everything clear on that? I'll take that as a yes. Okay, anything else? All right, if anything comes up, put it in the chat. What I want to do now is make sure that breakout rooms actually work. I don't know if they do. So we're going to try them now. And what I would like you to do is head over. See how this works. Head over to the class website and take a look at this lecture review. So we'll enter a breakout room, and I'd like you to talk to the people who are in there with you, whoever that may be, and reason through these. We have some time. So these post tests, as they're going to be called, these are here for you to check your understanding, and that's it. So after the lecture, or I guess after our class meeting, or during it, if we kind of have time like we do now, I'd like you to chat with each other and try to answer these, and then we can go through these together. So this will be, I hope, a good study tool for you when you're prepping for the exam. So I'd like you to make good use of these. Let's go ahead and try to make use of these now. And we'll see if this works. One let's plan on going through these in about, I don't know, I'll check back in 10 minutes or something like that. I don't know how long this will take. You never know. And I'm assuming you can still message me. I've never used breakout rooms, but just holler at me and let me know how things are going. All right. See what happens. Excellent. All right. Oh, hey, you can pause this. And we're all back, I think. All right. Why don't we run through this then? Okay. So I hope you got your name. All right. Here's a little electron micrograph image showing us a synapse, believe it or not. And turns out this is a chemical synapse. I did label the answer wrong. I caught that after a few of you submitted. My bad. We know this is a chemical synapse. Why do we know this is a chemical synapse? What are we seeing here?
The presynaptic vesicles.
Perfect. What arrow is pointing to some presynaptic vesicles?
Arrow C.
Thank you. Yeah. C. Look at all these little blobs right here. Dead giveaway that we have a presynaptic site. And if you look just across from it, notice this very dark area here. Why is it so dark?
Because it's dense.
Exactly. It's dense. Yeah, it's electron dense. It absorbed a whole bunch of electrons because it's filled up with proteins. We've collected a bunch of protein right here. So that's going to be our postsynaptic density. Here's the presynaptic site. Notice there's not a whole lot of space between these. They are right there next to each other. That's going to ensure that as soon as we spit out our neurotransmitter, it's going to contact neurotransmitter receptors. We're not going to have to wait. Now, you might think that it's A, but it's not. That's a mitochondrion. We can see another one down here, another one down here, another one over here. They're all over the place because you need ATP all over the place.
But how do you know that that's a mitochondrion? Like, how do you, like, how do you know?
Great question. So how in the world can I tell? Magic. How many, did this just zoom for you all? I can't see you. Okay, so it didn't zoom. Okay, no problem. Do whatever you need to do on your screen to zoom in, but what I want you to do is count how many membranes do you see on this thing? We got an outer one, and then there's one right inside of that. Do you see that? You see the double membrane there? It's so beautiful. It's like a double rainbow, but it's a double membrane. You should know that a mitochondrion is going to have two membranes. So it probably started as its own little organism and they got engulfed. And it was surrounded by another layer of membrane and enslaved by the eukaryotic cell that took it up. It's got a nice place to live, but really when you think about it, it's doing all the hard work. So that double layer there, that's a dead giveaway, mitochondria. And then there's all this other crap in there. Now that's just because we've sliced through the membrane. This inner membrane is quite wrinkly. And so what we're just seeing are those invaginations, but we're looking at a three-dimensional structure in 2 dimensions. So that's the dead giveaway. Great question. Will you ever have to interpret an electron micrograph? I don't know. Probably not. But you should definitely be able to pick out the presynaptic site. That thing is fairly important for a neurobiology class. And what you're going to see is a whole bunch of vesicles all throughout there. And I could draw it, but you have a real picture right there. And you'll notice it's filled up with vesicles. And each one of those is filled up with neurotransmitter. So that's the presynaptic site. And we know this is a chemical synapse, not an electrical one. I realize I put that first. I'm an idiot. I make mistakes. We know it's a chemical synapse because we see all those vesicles. It has to be a chemical synapse. There's the chemical right there. It's in these little packets of membrane. Okay, microglia. We've heard of these before. We heard of these last semester. You're not allowed to forget last semester. So you got to remember microglia. Are they going to act as macrophages? Absolutely. They're going to engulf stuff, whether it be a bit of cell debris or some foreign invader. They're going to eat it. Are they going to release pro-inflammatory cytokines? Yes, they are. I think that was lecture 2 last semester. Revisit that one. It's a good one. Do they respond to injury and infection? Absolutely. So even if you didn't remember pro-inflammatory cytokines, you said, what the hell is that? You should remember macrophage, respond to injury and infection. It's got to be all of the above. Microglia are going to do all of that. And more, but we'll forget about the more. All right, so if you have a complex task like motor learning, which ain't easy, it's just like pimping. If you're trying to learn how to throw, let's say, a triple 20 in darts, that's tough. You have to learn a fairly precise set of movements. You got to consider how far am I from that? How many beers have I had? You got to take all that into account whenever you throw your dart. It's complicated, but we can learn that. I've seen some super drunk people throw some really good darts. And the way that they can learn that is by having really complicated neurons dedicated to this really complicated task. So really what we're asking here is, what's the most complicated neuron? Which one can take into account the most information? Let's think about what these different neurons are going to look like. Bipolar neuron, pretty simple. It's got a dendrite, it's got an axon. That dendrite is usually quite small. Let's think about a multipolar neuron. Multiple dendrites. Remember, most of your synapses are going to be axodendritic. A lot more sites for synaptic input, so they can take into account a lot more information. Pseudo-unipolar or unipolar, doesn't matter. No true dendrites. We got an axon out here that has some specialization. So here's your dendrites, just a little bit out here. These are going to be your sensory neurons. So those neurons that live in the dorsal root ganglia, they start out as bipolar cells. They fuse their axon and dendrite together. And all they're going to do is notice is someone touching me right here. Over here, I don't care, right? Just right here. So fairly simple information because they have fairly simple dendrites. But something like a multipolar neuron, or it doesn't have to be pyramidal, it could be a more complicated Purkinje cell that's going to have this elaborate dendritic armor up here, a little bit down here. The more dendrite you have, the more synapses you can have, the more information you can consider. So we should think about multipolar neurons here. We're going to tend to see bipolar and pseudo-unipolar neurons as simple relays. Those are going to be sensory neurons like you see in your retina for bipolar cells, or your spinal cord for pseudo-unipolar cells. But when you get into the brain where the magic happens, multipolar. Let's see. Okay, we're trying to describe the function of the axon. So conduct chemical messages from the cell body to the presynaptic site. Probably not. Well, let me see what, yeah, let's see what we picked. If you got this, then I'm not gonna, belabor it. Okay, I'm not gonna belabor that. I didn't need to do the last one either. Okay, we do need to talk about this one because you fell for it. You picked all of the above. What the hell? Why wouldn't you? Why would I write that if it weren't true? So neurons connected by electrical synapses. They do form gap junctions, but that doesn't mean that they leave a little gap between their membrane. They actually fuse their membrane. The gap junction creates gaps within the membrane. So if you actually look at a gap junction, the membrane itself is fused. Something like that. So here's cell 1, the inside. Here's cell 2. Here's your gap junctions. These are going to be lined. There's going to be little pores. It's not just a hole. It's going to be little channels that are created. But the membranes themselves will actually fuse together. And we're going to share our cytoplasm. This will not affect the amplitude of the action potential. It won't be any stronger. It has nothing to do with that. What it does is synchronize their activity. So if one is active, the other tends to be active as well. But action potentials are all or none. They either happen or they don't. So it's not about changing the strength of the action potential. It's going to change the strength of the chemical message that they release, but only because we have additional neurons recruited. Volume transmission, we should be thinking mono amines. We're doing that. We got our ependymal cells. Astrocytes do all of this. They are definitely connected to each other by gap junctions. That's how they form that network. So that if one is very close to a highly active area and picking up a whole bunch of glutamate, let's say, it can spread that out to its neighbors. So if I'm listening in on this synapse and it's very active, it's spitting out a whole bunch of glutamate, and I start to pick that up, when glutamate concentrations increase, it's going to be harder to pull it in because the diffusion gradient is not as strong. So what I do, send the glutamate out to my neighbors. I keep glutamate concentrations low here so I can still effectively remove it from the synapse. So we definitely need to pick that top one. Contact blood vessels, absolutely. We went through that on Monday, so we should know that. So it's got to be all of the above. We just selected 2 of them. The third one's also true. You get the point. Okay, we got to look at a figure. this is not an uncommon task for me to ask you to do. So look at a figure. Let's have a look. It's good to be able to look at pictures and make sense of them. Alrighty, so this figure is showing the number of extensions or the little branches that come off of microglia. So you should hopefully remember that the microglia are going to kind of look like little neurons. They're going to have these extensions coming off of them. But in response to injury, what they're going to do is kind of round up and adopt a more amoeboid shape. So they'll be kind of round, and that allows them to crawl more effectively. You don't move well when you have these intricate extensions. So they retract them, so they're a nice little ball, and then they move. It's much easier to squeeze around when you're a ball. We see them becoming reactive, in other words. And that should make sense because what we're doing here is adding lipopolysaccharide, which is a molecule that you find on bacteria. So what we're essentially doing is telling the microglia that there's bacteria around. So we put a bacterial surface marker, just a little lipid polysaccharide that's specific to the bacteria. The microglia think there's a bacterium. That's, of course, going to make them reactive. So all we're seeing here is the rounding up of microglia, and you can see that here. Just compare this little one here and see to that, or you can look at the bar graph. They do the quantification for you right there. Let me see what this does. Might do nothing. All right, it does a little something. Does a little more something. Cool. So the bar drops. The number of branches decreases. In other words, it rounds up. So it's becoming reactive. That's what we want to see here. We're definitely not seeing them die. We don't see them eat anything. I just pulled this out of my ass. So formation of reactive microglia. Okay, so which of the glial cells are not derived from nervous tissue? Microglia. Everything else is going to be derived from neural precursors. So microglia are going to kind of crawl their way in early in development from the yolk sac. Astrocytes are derived from neuroprogenitor cells, for example, radioglia, later on in development. So they'll come after neurons. Oligodendrocytes, also from glial precursors. You might be tempted to pick Schwann cells because they're in the peripheral nervous system, but they're still going to be derived from neural crest cells, nervous tissue. So neural crest cells are going to give rise not only to the neurons, but also the glia in the peripheral nervous system. The one that we want to pick here is the microglia. These are not nerve cells per se because they're not derived from neural precursors. Okay, you probably got volume transmission down. I guess I need to take a look. Go. Let's see where we're at. Okay. We want to think monoaminergic for volume transmission. Let's see. CSF. Okay, we got our ependymal cells. Wait, I already did this. Where am I? Okay. We're back. Okay, in volume transmission, the site of neurotransmitter release is not near the postsynaptic site. So retrograde transmission just means that we're sending something back. So the dendrite, for example, is going to release a transmitter that's picked up at the presynaptic site. They're still right there next to each other. Okay, we got another image to look at and another image to look at, and we definitely need to look at the last one. The first one, it looks like we're getting it, so electrical synapses. But let's take a look and make sure that makes sense. Okay, so I think it's always good to look at actual data and try to get a sense of, is this real? So what we're looking at here, in part A, this is just showing you what they're recording from. And here's a cartoon schematic. So we got a couple of cells, and look, they're putting their cell bodies right next to each other. That's because they're fusing their membranes. We can tell that they're fusing their membranes because of panel B. Let's look at panel B. So we're recording from both cells at the same time. We're going to deliver current to one of those cells. So cell 1, we're going to deliver depolarizing and hyperpolarizing potentials. and then we're going to measure the currents in both cells. We're not doing anything to the second cell. So we're not delivering any potentials, but we see currents. So whenever we deliver a potential to the first cell, we get a current in the second. When it's hyperpolarizing, we hyperpolarize. When it's depolarizing, we depolarize. Pretty nifty, and it's immediate. So could this be explained by electrical synapses? Absolutely. Could it be explained by GABA release from the left cell? No, GABA is not going to depolarize us. How about glutamate release? No, glutamate's not going to hyperpolarize us. This is only explained by electrical synapses. Please don't just pick all of the above. I know that trick. This is the one we probably need to look at. So we haven't covered Ohm's law, although I did mention it earlier. But it's V equals IR. And you really need to commit this one to memory. It'll explain pretty much everything we talk about whenever we deal with the electrical activity in neurons. It's all about Ohm's law. Okay. So let's see. Voltage, that's the V. Charge buildup is the product of current and resistance. So that if you're moving charge and then you're resisting its movement, it's going to build up into that area you moved it into. So let's take a look at this. In panel A, we can see some calcium imaging. A little bit of calcium here, a whole lot more here. All right. Who cares about that? Good thing I said that. Okay, below that we see currents that are measured in astrocytes. And these currents are brought on by applying different potentials. So we apply a potential. This is just like what we saw before. Apply potential, measure a current. Look at how the current changes. So we're applying the same potentials, we're getting less current. Okay, so like I said, pay attention to the currents on the right. So when we add octanol here, we're seeing some sort of change in the currents. Are we getting more or less current? Let's just go ahead and get that out of the way.
Less current.
Thank you. So the current amplitude is reduced. We can clearly see that. These move a lot further from baseline than these. These are all kind of squished together. What's Ohm's law?
V equals IR.
Thank you. So this should be a reflex. Kind of like you tap a tendon, stretch the muscle, cause contraction. When you hear Ohm's law, you should think V equals IR. Same thing. Still Ohm's law. Still applies here. Our currents have dropped. Our potential steps have stayed the same. We haven't changed that at all. We're still applying the same potentials. We're going from minus 80 up to 0 millivolts or down to negative 140 millivolts. And the current is now decreased. Well, we haven't changed our potential, so let's not think about that. What could cause this?
A change in the resistance.
Exactly. An increase in our resistance. So it's much harder for current to move here. All right, so we're doing this in astrocytes. Yeah, I think I wrote that. So what would make sense for decreasing the resistance? I'm sorry, increasing the resistance here? Now remember, if this cell picks up a whole bunch of neurotransmitter, it can spread that to its neighbors. It's going to spread through gap junctions. So all of these astrocytes are coupled together and they essentially act as one cell. They share that cytoplasm. Stuff can move from one cell to another. Resistance is the opposite of that. Resistance is impeding the movement of stuff. So whenever we apply a current, for example, so let's say we patch onto here, we got this is a glass pipette with an electrode in it. And when we apply a potential, the amount of current that we get is going to depend on how much resistance is there. When there's higher resistance, we get less of a current. When there's less resistance, such as when we have a whole bunch of gap junctions connecting us to our neighboring cells, we get more current. So what octanol is doing, you could have probably Googled it, I'm sure, it's blocking gap junctions. So now what you're doing is isolating just a single cell here. You're blocking its connections with its neighbors. And so now what you've done is cut off avenues for current to move. In other words, you've increased resistance, which is what would explain the decrease in current. This is to say that astrocytes do have gap junctions. They do have a means to share their cytoplasm with each other. So they're going to share all kinds of things. In this case, we're seeing them share charge. Up here, we're seeing them essentially share calcium. When that calcium doesn't have anywhere to go, it builds up a whole lot more in this astrocyte. If the neurotransmitter didn't have anywhere to go, it would build up more in this astrocyte. So gap junctions are pretty important for how astrocytes work. Okay, any questions on that stuff?
I have a question about that last one.
Sure.
So, like, the other options were also like just different types of holes in a membrane, whether they're transporters and stuff. And if you block the holes in a membrane, wouldn't it also like increase resistance, albeit like in a different way? So why are they wrong?
It could. Notice that all of those say transporters. So there's a big difference between a channel, a transporter, and a pump. So if we have an opening in the membrane, where are we at? There we are. So if we have an opening in the membrane, that's what a channel is. Oh, that's the wrong one. So channels are going to form little pores in the membrane that stuff can freely move through in either direction. That's what a gap junction is as well. So let's say the channels are going to be able to move somewhere around, let's see, what is that? What is that value? Somewhere on the order of 100 million or so ions per second. Right? About 100 million. Now, a transporter, on the other hand, is going to have not a hole in the membrane, but it's going to have specific binding sites for different molecules, and it's going to have to change its shape to move stuff across the membrane. So a transporter doesn't make a hole. It makes a pocket. And it can move stuff, but it has to change its shape in order to allow stuff to move each step of the way. So we can move about 1,000-fold fewer ions per second. If we go down to pumps, now we're on the order of about 100 ions per second that we can move. So we're taking about 1,000-fold step reduction between channels to transporters and transporters to pumps. Pumps have to spend ATP to change their shape. Transporters are going to ride along those passive ion diffusion gradients. So they're a lot faster than pumps, but they're a lot slower than channels. So when you're applying a potential, that's not really going to have much to do with whether this transporter works or not, but it is going to affect how charge moves through channels. If, on the other hand, we were, let's say, applying glucose, that's not spelled like that. If we're applying glucose, well, that would affect the movement through transporters. Or if we were applying GABA, but the potential change isn't going to affect transporter currents. Transporter currents need the thing that they're moving. So unless we were, well, no. What you'd see with the transporters is, I need to share. Hold on. This is not a burden at all. Okay. All of this would change. So there'd be a change in the current all the time, and it would have nothing to do with potential. When you see currents by delivering a potential, you should be thinking channels. Transporters just move stuff when they're there. So it could have been any of those, but in this case, it's gap junctions. And it's gap junctions because we're actually getting an appreciable current with our potential changes. Transporter currents are very hard to measure. You just wouldn't know that unless you took this and got it wrong, and then I told you, and then now you know it. But you should know that we have those 3 orders of magnitude difference between channels and transporters and pumps. I think this is in the notes. Might not be this one. Coming attractions, I suppose. The good news is you take this quiz time and time again and make sure that that sticks. And there's nothing more informative than getting a question wrong. Any other questions? All right, well, if they come up, you know my email address. If you're in town, you know where I sit. I'll see you Friday, and we'll go through development, I do believe. And then next week, we get into the good stuff. But not on Monday, right? Okay, cool. Well, have a great rest of the day, and tomorrow, I'll see you Friday.